Good morning, good afternoon, and good evening, everyone, and welcome to our webinar, Studying Epilepsy in Awake Head-Fixed Mice Using Microscopy, Electrophysiology, and Optogenetics. This is Liam Sanyo from Insight Scientific, and I'm very pleased to be your host for today's event. Our session is sponsored by Neurotar and will feature Dr. Rob Wykes, Senior Research Fellow at UCL Queen Square Institute of Neurology and Senior Lecturer at the University of Manchester, and Dr. Anthony Umpierre, Postdoctoral Researcher at the Mayo Clinic. Dr. Wykes and Dr. Umpierre will discuss how microscopy, electrophysiology, and optogenetics can be used to study microglial calcium signaling and epileptic networks in awake head-fixed mice. And with that, I'm very pleased to welcome our first presenter, Dr. Rob Wykes. Rob, thanks so much for joining us today, and feel free to take it away whenever you're ready.
Okay, well, hi, and thank you very much for giving me this opportunity to discuss some of our in vivo epilepsy research in awake mice. In this webinar, I will briefly discuss why I believe it's important to be able to do recordings in awake brain. I'll then discuss some calcium imaging research that I started several years ago now as an Epilepsy Research UK Fellow, work that I conducted with Federico Rossi in the lab of Matteo Carandini. And then I'll move to discuss some more recent work using graphene microtransistors and calcium imaging approaches that are ongoing in my research labs at UCL and at the University of Manchester. In order to study and understand networks, whether local or long range, it is important to record from intact brain. Traditionally, in vivo imaging and multi-channel electrophysiological recordings have been made under anesthesia in rodents. Although informative, confounders in these, in particular those associated with anesthesia, limit the value of these experiments. In recent years, efforts have been made to improve and optimize experimental setups to permit recordings in awake head-fixed rodents. Imaging and electrophysiological data obtained in non-anesthetized animals are more relevant and allow behavioral tests and observations to be included in the experimental paradigm. Optogenetics can be used either to stimulate or inhibit populations of cells. Electrophysiological methods can be used to record brain activity over multiple frequencies, and due in a large part to rapid improvements in genetically encoded indicators of neuronal activity and in microscopy, calcium imaging is providing novel insights into seizure initiation, propagation, and termination. A small but growing number of labs are using these techniques, either individually or in combination, to gain a better understanding of the cellular and network changes that occur during acute seizures and in chronic epilepsy. My lab has developed and optimized a recording setup that permits mesoscopic calcium imaging, full bandwidth multi-channel recordings using novel graphene-based electrophysiological probes, and the ability to on-demand optogenetically induce cortical spreading depressions. We are using these approaches to gain a better understanding of seizures and spreading depolarizations in awake mice. When I first started in this area about 6 or 7 years ago, we used a styrofoam ball to support the awake mouse whilst head fixed. More recently, we have switched to using an airlifted platform, which offers several practical advantages during complicated experiments, which require access to the brain for multiple stimulating or recording probes, as well as multiple cameras, which allow mesoscopic calcium imaging, eye pupil tracking, and also cameras for behavioral activities. We start by surgery to perform and attach head bars to the mice. After full recovery from this surgery and prior to experimentation, animals are acclimatized to the head fixation chamber. We perform multiple rounds of habituation, starting with a 15-minute session, and then increase the duration over several sessions to a final habituation that lasts an hour or longer, and where the animals show no or little signs of stress or anxiety. To minimize the amount of time it takes a researcher to perform these habituations, we now do this in batches of 3 animals. Epilepsy research is rapidly adopting novel fluorescent optical imaging methods to tackle unresolved questions on the cellular and circuit mechanisms of seizure generation and evolution. Fluorescent markers can be targeted to neurons to read their activity. Reporters change the intensity or wavelength of their fluorescence as a function of neuronal activity. Fluorescent reporters targeted to neurons at the population level read out the hallmarks of epileptic activity. During normal processing, neuronal activity is sparse and EEG is desynchronized. During seizures, activity escalates, recruiting most of the neurons in the network and generates hypersynchronous EEG oscillations. Two-photon imaging can be used to record neuronal activity with single-cell resolution in a small cortical volume. In contrast, wide-field imaging can be used to record neuronal activity over the entire dorsal cortex of rodents. Two-photon microscopy can record single neuron dynamics during interictal and ictal discharges and highlight the differences between the activity of excitatory neurons shown in green and inhibitory neurons shown in magenta. They can also look at how these neuronal classes are involved in the initiation and propagation in the immediate penumbra. Wide-field fluorescence imaging provides mesoscopic mapping of the functional anatomy of the cortex. It can investigate seizure propagation pathways and ask how the unfolding of epileptic events depends on the topology of the brain-wide functional connectivity. We used a mesoscopic calcium imaging approach. We targeted an LFP pipette into the visual cortex. The pipette solution contains 10 millimolar picrotoxin, a GABA receptor antagonist. Local diffusion of picrotoxin from the pipette tip induced epileptiform activity, which induced interictal spikes and seizures, recorded electrographically from the LFP pipette and detected by large changes in GCAMP fluorescence. A rhythmic pattern of interictal spikes, shown by the blue triangles, and seizures, shown by the red triangles, evolves. In addition to a correlation between calcium transients, green, and the LFP, shown in black, eye pupil dilation, shown in orange, and increased running, shown in yellow, are correlated with seizures. The mouse visual cortex offers a simple substrate to systematically study the contribution of both local and long-range connections to the propagation of epileptiform discharges. The functional connectivity within and across visual areas can easily be mapped. Higher visual areas inherit their retinotopic representation from V1. For example, the neurons in the red area of LM are functionally connected to the neurons in the red area of primary visual cortex. For each animal, we determined a retinotopic map of primary and higher visual areas by preventing contrast-reversing grating visual stimulation on LCD monitors. After determining a retinotopic map for each animal, we then investigated the spatial temporary evolution of interictal and ictal discharges and related these to the functional connectivity underlying normal processing in the awake visual cortex. In particular, we asked whether seizures preferentially follow contiguous or homotopic propagation pathways. In this video, you will see on the right side 3 circles. The black circle represents the seizure focus in V1. The green circle represents an area of higher visual cortex that inherits its retinotopic preference from the area in V1 represented by the black circle. The red circle represents an area of V1 with a distinct retinotopic preference, but which is physically closer to the seizure focus. In the left-hand panels, you see the LFP signal in blue, and below are the changes in calcium detected from the 3 circles. At seizure onset, there is an initial increase in fluorescence within a spatially restricted area of V1. This can be well modeled as a standing wave. The seizure then starts to propagate as a traveling wave with both contiguous and homotopic components. To quantify this, we drew a line from the seizure focus to a higher visual area with a shared retinotopic preference. This line contains multiple regions of interest, each color-coded with its own retinotopic preference. As you can see, 6 to 8 seconds into the seizure, the ictal activity shows a bimodal profile, being stronger in the homotopic regions in LM than in the regions of V1 that are closer to the focus. The average propagation speed was calculated to be about 0.5 millimeters per second. These results indicate that the delay to ictal invasion depends approximately on 2 factors and follows both contiguous and homotopic propagation. Once this territory was invaded by a seizure activity, it displayed striking oscillations in the range of 6 to 11 Hz, evident both in the LFP and in the GCAMP fluorescence. The seizure focus acted as a pacemaker for these oscillations, and homeotopic regions oscillated almost in synchrony with the activity of the focus. However, once the seizure spread beyond the visual cortex, more complicated patterns of oscillation emerged. In approximately about 30% of these seizures, we observed at the end a postictal cortical spreading depression. The black line indicates the LFP at seizure focus. The red line is the GCAMP6F signal averaged across the whole imaging window. The vertical green line indicates the start of the cortical spreading depression. Single frame snippets from the GCAMP6F movie correspond to the black dots. The cartoon electrode indicates the site of picrotoxin injection and the LFP recording. The CSD wave started at a site distinct from the seizure focus, and the depolarization spread radially with no apparent relation to the functional organization of the cortex. These CSDs propagated at around 3 to 5 millimeters per minute. The calcium imaging movies demonstrate that diseases like epilepsy have neurological activity over a wide range of frequencies. Current electrode technology is not well suited to recording infraslow activity, such as that observed during spreading depolarizations. In contrast to standard passive electrodes, graphene transistors have full bandwidth recording capabilities, resulting from the direct field effect coupling of the active transistor, as well as from the electrochemical inertness of graphene. They come in two varieties, either a 16-channel epidural grid or a 14-channel penetrating probe. The electrophysiological signal measured by the graphene transistors is acquired by a specialized amplifier made by GTEC, which enables simultaneous recording in two frequency bands with different gains, therefore preventing amplifier saturation. In contrast to AC-coupled or DC-coupled passive electrodes, which encounter baseline drift and require either high-pass filtering or electronic offset readjustments, graphene transistors are able to record infraslow activity, including spreading depolarizations, over several hours with minimal baseline drift. To demonstrate their capabilities to detect infraslow activity, we injected potassium chloride, a known trigger for cortical spreading depression, into the motor cortex and detected the propagation of this depolarization as it passed over somatosensory cortex using an epidural graphene transistor array. Each line represents the cortical spreading depression recorded from an individual transistor in the array. Note the spatial temporal resolution capable using this approach. In this example, I'm showing you a seizure and spreading depolarization recorded using a penetrating probe inserted through visual cortex to the hippocampus. The chemical convulsant was injected into the cortex, and a DC shift at seizure onset is observed. A postictal spreading depolarization is first detected in the hippocampal channel. Can we use calcium imaging and graphene microtransistor technology to investigate mechanisms underlying seizure onset? Our calcium imaging data indicates that the spatial temporal footprint of an intraictal spike is exactly the same as the first couple hundred milliseconds of a seizure, suggesting, at least in this model, that some intraictal spikes can transition into seizure. Again, if we look at the trace, the blue arrows correlate with the intraictal spikes, and the red triangles are brief seizures. This asks the question, what is special about the intraictal spikes that do transition into seizure? Is it a failure of inhibitory surround? Or are there some slow accumulation of excitatory processes which promote a subsequent intraictal spike to evolve into seizure? We can separate our full-band graphene transistor recordings, either high-pass filtered above 0.5 Hz or low-pass filtered below 0.1 Hz. When we do this, we see that prior to seizure initiation, there is an appreciable DC shift which grows in amplitude at seizure onset, supporting the hypothesis that the brain network is changing and slow processes are occurring prior to seizure onset. This supports modeling and potassium-sensitive electrode recordings previously reported by other groups. By using multiple arrays, we can examine seizure transition across brain regions. In this example, an epidural array is placed over the visual cortex, and a penetrating array through the cortex and into the hippocampus. Chemiconvulsant is injected into the superficial layers of the cortex, and this soon generates a low-frequency 2 Hz rhythm. In contrast, deeper layers of the cortex and the hippocampus are relatively quiet at this time point. Upon seizure, activity dramatically increases in the hippocampus, and a spreading depolarization then silences activity in that region, as well as deeper layers of the cortex, but does not stop the rhythmic 2 Hz activity in the superficial cortex. If we take a closer look at the hippocampal channel, we can see that there is a marked change in activity halfway through the seizure that coincides with high gamma 80 to 200 Hz activity, indicative of hippocampal recruitment into the seizure. This is shown by the time indicated by number 4 in the blow-up. If we perform phase amplitude coupling between an epidural cortical channel and a channel located in the hippocampus, we detect strong coupling at the time point where the hippocampal recording transitions from higher amplitude, lower frequency firing to lower amplitude, higher frequency firing, and also observed at the same time point as the increase in high gamma activity. The low frequency activity in the cortex appears to modulate the high frequency activity in the hippocampus to a much greater extent than the low frequency hippocampal activity modulates the high frequency activity in the cortex. The strength of this coupling is transient, lasting only a few seconds, and could provide insight into how seizures transition across brain regions. Finally, as CSDs are associated with worsening outcomes in several neurological disorders, including stroke, migraine, and epilepsy, they are a potential therapeutic target. However, translational success of CSD-suppressing drugs has not been particularly effective. A major reason for this may be that a large number of the preclinical studies have been performed in an anesthetized brain. Therefore, we sought to adapt our in vivo awake preparation to allow a robust platform for drug screening. To do this, we expressed channel rhodopsin in the motor cortex to allow optogenetic induction of CSD and record its propagation using an epidural graphene transistor. Blue light evokes a CSD, the black trace shows the full bandwidth recording of the depolarization, and the red trace shows the high-pass filter signal, clearly visualizing activity suppression during the cortical spreading depression. This approach is extremely robust and reliably induces CSDs, displayed as a 90-minute recording with CSDs elicited every 10 minutes. Note the minimum voltage drift over 90 minutes. Therefore, we have a robust platform to be able to do a drug screening analysis. As a proof of principle, we can inhibit CSDs with MK-801, an NMDA antagonist. Although MK-801 is not a clinically relevant drug due to side effects, we believe this screening approach in awake brain could be used to identify clinically relevant drugs in the future. In summary, I'd hope to have given you a taste for my lab's research using calcium imaging, full bandwidth electrophysiology, and optogenetics in awake head-fixed mouse to investigate seizure dynamics, and in particular the role of spreading depolarizations in epileptic brain. There are many people to thank. The calcium imaging work was conducted with Federico Rossi. Numerous people have contributed to the graphene transistor recordings in my lab. In particular, I'd like to flag out Martin Smith, Edu Masvidal, and Andrea Bonaccini. And the arrays were designed and fabricated by 2 labs in Barcelona, led by Jose Garrido and Anton Guilmero Brunet. Finally, I'd like to thank my funders and you for listening. Thank you.
All right, thank you so much, Rob, for the fantastic presentation. And with that, I'm very pleased to introduce our next presenter, Dr. Anthony Umpierre. Tony, thanks so much for being with us, and feel free to take it away whenever you're ready.
All right, thank you, Liam, and thanks to the team at Inside Scientific for preparing this webinar. I'd also like to first thank Elena and the team at Neurotar for the invitation. Today, I'd like to tell you a little bit about my postdoctoral research in the lab of Longjun Wu at the Mayo Clinic. What we're doing is investigating the impact of neuronal activity shifts on microglial calcium activity in the awake head-fixed animal. For those of you joining from a background studying neurons, let me briefly introduce microglia. Microglia are the innate immune cell of the central nervous system, And through their immune roles, there have been a number of documented examples about how microglia can be both reparative to brain tissue or potentially increase damage through their pro-inflammatory actions. In this field, it's thought that a lot of the functions of microglia really begin with their dynamic processes, which can be motile and respond to injury within a few minutes of damage occurring. These cells are also highly attuned to inflammation, and as the topic of today's webinar, these cells are also, we're increasingly finding, sensitive to neuronal activity shifts. So in the next few slides, I will show you a few key examples of how microglia structurally respond to neuronal activity changes, including first hyperactivity. In this video, green microglia labeled with GFP are shown both before and then after topical application of 1 millimolar glutamate. And what you might be able to appreciate are these small filopodia-like microglial processes that pretty rapidly extend after glutamate application. And what they do is they actually preferentially move towards these red Thi1 labeled neurons of the hippocampus, making pretty robust contact. What we find is that this is a pretty rapid response occurring within about 5 minutes of glutamate application. And this work was done by a previous postdoc in the lab, AO. On the other end of the activity spectrum, what was probably most surprising is Yang, another postdoc, found that if you introduce isoflurane anesthesia, an anesthetic, to an otherwise awake animal and introduce periods of neuronal hypoactivity, what you see is actually a pretty similar response where microglia begin extending their dynamic processes, almost searching through the brain for something that seems to have been disturbed, something that actually evokes structural responses. In between these two studies, we have our first indication that microglia are actually responsive to bidirectional shifts in neuronal activity and engage structural responses. When I joined the lab, I was interested in how intracellular signaling pathways in microglia, namely their calcium activity, might co-occur in relation to their structural changes. And I felt that understanding this dynamic of microglia and a functional lens could inform us better about how microglia respond to their extracellular environment. And one of the first things we did was we began with the best approaches to study microglia calcium in the awake animal. And if you study microglia, you might well know that viruses are not often efficacious in transfecting microglia. So we're often left with using genetically engineered mice to accomplish our task. In this case, I use the GCamp6S mouse, which is Cre-dependent, and I breed this to a microglial Cre line, which is the CX3CR1, also known as the fractalkine Cre-ER line. And after administering tamoxifen to the animal, I perform a craniotomy and place a cranial window over the somatosensory cortex. And I also secure this area with the Neurotar 4-point restraint. And this allows us to perform 15, 20, or even up to 90-minute imaging of the mouse in a single Z-plane with pretty good Z resolution, which is really essential for getting a good quality time series. Now, before I jump to the next slide, I wanted to mention what really sets microglia calcium apart from other cells in the brain, and that is that microglia have very low, almost absent, spontaneous calcium activity, as reported by a number of other in vivo studies. However, when we did our awake preparation, what we first noticed 24 hours after implantation, as shown here, is that in this acute window, 24 hours after surgery, we actually saw a lot of spontaneous calcium activity. However, in this chronic window, 1 month later, that predominantly processed calcium activity has largely reduced, almost by about half. So for those who want to study microglia calcium, I wanted to make this a first technical note. The acute window preparation, 24 or 48 hours after surgery, often has very high spontaneous activity. However, a month later in a chronic window, that predominantly processed spontaneous activity has reduced. Another great thing about chronic imaging is that you can fully train the animal to be habituated to the head fixation as shown in the top. We perform a number of platform training sessions as shown very well by Rob's videos that allow the animal to be fully acclimated to the environment and the head restraint. Part of the reason we felt that the acute window did not accurately represent spontaneous microglia signaling is because a number of studies have shown that microglia calcium activity is pretty low at rest. It's pretty low in the basal state. However, a number of different pathological features can evoke greater activity, such as proximity of microglia to amyloid beta plaques, neuronal damage, millimolar ATP application, a bacterial-like infection simulated by LPS, or a focal laser burn. All of these events can take the low microglial calcium activity at rest and elevate it significantly. But on the other hand of the spectrum, we weren't exactly sure what neuronal activity did to microglial calcium. 2 studies using bicuculline, the GABA antagonist, to elevate neuronal activity came to opposite conclusions. In one study, the application of bicuculline alone had no effect on microglia calcium, despite the fact that they could show neuronal activity increases. On the other hand, a study showed robust increases in microglia calcium following bicuculline application. But in this study, it's important to know that the activity increases were evoked in the presence of LPS and the inflammatory context that that provides. So for me, one of the important early questions was really, how does neuronal activity sort of comprehensively alter microglia calcium? And to do that, we began with an approach that includes activity shifts, both pharmacological and chemogenetic tools. So what we did was we used pharmacology, including anesthesia and kainate, to evoke seizures on the 2 extreme ends of neuronal activity. And we also complemented those studies with both GI and GQ chemogenetic dread approaches to change activity to lesser degrees. We know from previous work in the lab that the structural response of microglia is to engage process extension when neuronal activity shifts from a baseline state to a hypoactive or a hyperactive state. And I hypothesized that largely structure would follow function and vice versa, and that under conditions in which microglia began dynamic process extension, I hypothesized you would also see the greatest calcium activity. This is also based upon previous work in microglia culture preps, where calcium was shown to be an important effector in how microglia began process transitions and other larger motility events. So, towards our first experiments, what we did was, in the chronic window prep and using the awake animal, we increased neuronal activity, in this case using kainic acid, an AMPA and kainate receptor, and the levels of kainate that we applied to the animal were pathological. They were specifically used to evoke seizures. In all future slides, what I'll do is just quickly show you a video that shows how the activity changes from baseline to after the application of a drug or chemogenetic agent. But in this first experiment, what I wanted to show you was a little bit about our experimental details. So what we're doing is we're performing time series. So videos from a single Z-plane, they're often 15 minutes long and imaged at 1 Hertz. This speed is actually sufficient to capture most microglial calcium activity because these transients are typically slower, on the order of 5 to 10 seconds or longer. We're largely imaging microglia in layer 1, 50 to 70 microns below the dural surface. And again, these animals are awake unless specifically we're using isoflurane to decrease neuronal activity. What we do is we compare the calcium activity from cells within subjects. So we compare the cell's calcium activity at baseline and then after an intervention, such as kainate is applied to the animal. And what we're most specifically interested in is domains or subregions of the cell, specifically the soma versus the process. And this is because in another glial cell type, the astrocyte, somatic calcium signaling is far less frequent than process calcium signaling. And in microglia, the processes are also the most dynamic. So when we evaluate microdomains, in this case after kainate application, what we find is there's a trend but not a significant change in somatic calcium activity on the left in these bar graphs. In future, somas will be shown in green and processes will be shown in purple. On the other hand, these purple processes of microglia, they show about a 3-fold increase in their calcium activity during acute seizure activity in the kainate model. So this was our first evidence that there were microdomain differences in how these cells engage process activity with calcium. Now, if we flip the script and strongly decrease neuronal activity, in this case using isoflurane to evoke general anesthetic states, we also looked at microglial calcium signaling. And in this experiment, we have the awake baseline, and then we introduce isoflurane. So actually the animal, this is another nice feature of the Neurotar system, we can keep the animal on the platform the whole time and just introduce the nose cone to this awake animal, evoking both anesthetic induction and then maintenance. And we can pretty clearly see the same cells and watch their calcium activity over about a 40-minute period. What we find in this line graph is that microglial processes about double their calcium activity 10 to 20 minutes after isoflurane is introduced to the animal. While on the other hand, somatic calcium activity in microglia is completely unaffected. So once again, we see that processes, these dynamic structural components of microglia, also have the most dynamic calcium shifts. And it's also important to note that unlike astrocytes and other cells that sometimes show very clear real-time calcium signal changes, microglia seem to show this 10 to 20 minute latency in how long it takes to evoke differential calcium activity. Now, while both kainate and isoflurane are great ways to change neuronal activity, they don't come without confounds. Isoflurane is also known to change the vascular tone of an animal as well as the metabolism, while kainate could certainly also engage some neuropathological processes and inflammation alongside its activity increases. So we wanted a more direct and straightforward way to change our local neuronal activity, and we settled upon DREAD or chemogenetic-based alterations. DREADs are designer receptors exclusively activated by designer drugs. And we introduced DREADs to CAM kinase 2 alpha excitatory neurons of the cortex. We did that with local AAV transfection. And on the left-hand side, when we introduced the GI DREAD construct into CAM kinase neurons and activate the DREAD specifically with the agonist CNO, we can induce hyperpolarization of the local neurons, leading to less neurotransmitter release. So GI DREAD is our way to decrease local excitatory neuron activity. On the other hand, introducing GQ DREAD into these CAM kinase 2 alpha neurons and then activating GQ DREAD with the ligand CNO increases the available presynaptic calcium levels in the bouton, allowing for greater neurotransmitter release. So this is our ability to increase local excitatory neuronal activity. And ultimately, of course, what we're interested in is how these local changes influence microglial calcium signaling. So in the next video, you'll see in pretty rapid succession microglia calcium signaling from 2 different mice. The first mouse will be an example from a GI DREAD mouse, and then you'll see an example of microglial calcium signaling in the GQ DREAD mouse. So first, in the GI DREAD mouse, we see microglial calcium activity at baseline, and now we see an increase in microglial calcium after neuronal activity depression. Now, here's the GQ mouse, and after GQ enhancement of neuronal activity, we also see microglia calcium increases. And I'll let that loop through, but I wanted to mention a couple interesting things. So of course, behind this microglial calcium signal, we perform these experiments also with neuronal calcium to characterize both the time course of neuronal effects and the effects of the dosages of CNO used. What we found overall was that neurons had their peak shift in activity about 30 to 45 minutes after CNO injection. And what's interesting is this latency reemerges. So microglial peak calcium responses are shifted 15 minutes later after the peak neuronal responses. So once again, microglia have this delayed onset calcium activity in response to neuronal shifts. And now on the bar graph on the left, what we see is both in the GI and GQ DREAD animals, different doses of CNO have dose-dependent effects on process calcium signaling. However, we didn't see any somatic calcium signaling differences across either GI or GQ dread or any CNO dose tested. So once again, processes seem to have the most dynamic calcium signaling. So in the last few minutes of this talk, I wanted to also mention some of our main longitudinal studies. So we had the acute studies really show us that there are bidirectional calcium responses in microglia to neuronal activity shifts. So microglia engage calcium both when neuronal activity decreases and when it increases. But we also wanted to perform longitudinal studies to see how basically long-term shifts in neuronal activity, as well as an inflammatory environment, could alter microglial calcium. To do this, we modeled the early and intermittent phases of acquired epilepsy development using systemic kainate acid injection. And so kainate represents basically an insult that could later lead animals to develop epilepsy or spontaneous recurrent seizures. This often takes weeks to months in mice and could take months to years in humans. But in this interim, where animals go through a transitionary period without seizures, this is called epileptogenesis and has been well documented to include changes in cell death, inflammation, and large shifts in genetic profiles. We image microglia over a 14-day period, mapping it onto the epileptogenesis timeframe. And the reason we did this is because we know that this period includes a number of different triggers for microglial calcium, including inflammation, cell death, and now neuronal activity changes. And so we wanted to know basically what features of microglial calcium emerged during this shift. So beginning one day after kainate status epilepticus, so the animal is not actively having seizures anymore, but this insult has occurred. 24 hours later, what we see is pretty decent elevations in microglial spontaneous, I guess you could call it, calcium signaling. But what really separates this activity pattern from all of the acute experiments is now the calcium activity includes both the soma and process microdomains of the microglia having strongly elevated signaling. And in fact, the elevations of microglia calcium after this one insult status epilepticus, these elevations can last for up to 10 days. And after 14 days, and in some cases 21 days, we see a pretty strong cessation of that increased calcium signal, returning to near baseline levels. And this suggests that largely, though prolonged, this increase in calcium can be more transient. What's also interesting, especially after Rob presented some of his beautiful work on spreading waves, is that throughout the entire 14-day period, we can see spreading waves of microglial calcium activity. It's rarer, it occurs maybe once or twice per animal, but we did see it in all animals. What's really interesting, although I didn't have some of Rob's really nice electrophysiological recording techniques to also understand the neuronal activity in these moments, what's interesting though is that in other studies, if you introduce inflammation with LPS, and then add bicuculline to the local cortex, you can also evoke microglial spreading calcium waves, suggesting that this could be a signature of both potentially the co-occurrence of inflammation and neuronal activity increases. And what's particularly interesting to me is that these calcium elevations in microglia could be sustained for up to 5, sometimes even 10 minutes in the awake mouse. And the last note I'll end on is in the microglial culture literature, there's this idea that calcium elevations in processes or in the soma can change how microglia motility occurs. But this really hadn't been evaluated in vivo yet. One thing we really noticed, particularly in these longitudinal kainate studies, was that we could see a really strong co-occurrence of microglia calcium activity with processes that extended, or in this case, converged. Those sort of correlations were pretty well temporally correlated as well. And when we went back into our acute data set, we looked at a number of instances, such as the GI dread or isoflurane decreases in neuronal activity, and the kainate and GQ increases in neuronal activity. And when we looked at microglia, what we found was that microglia that had processes that extended. In this subset of processes, the extending processes, shown in the green bars, these are the processes that often have the largest calcium transients and the highest proportion of processes showing calcium activity. So across all 4 different techniques, what we see in common is that extending or outgrowing processes have higher magnitude calcium activity and higher association with calcium activity. So with that, I just wanted to quickly summarize. In our acute studies, we find that bidirectional shifts in neuronal activity can engage microglial process activity. And this occurs after a brief, about a 10-minute latency. And in longitudinal studies, what we find is that both microglial processes and somata show very strong elevations in their calcium signaling that can last for about 10 days and roughly return to baseline levels, suggesting a transient but ultimately very profound alteration in how this cell type uses calcium signaling. And with that, I would like to thank the lab, my mentor, Dr. Longjun Wu, and our funding sources. And thank you for listening.
Now I'm just going to invite Rob and Tony back on the line. And as a reminder, if you do have a question for them, you can submit it using the Ask a Question panel. And so let's jump right in here. First question today is, and I think you both touched on this, but could you just clarify how you habituated the mice and how long does habituation take? And so Tony, maybe I'll direct this one to you first.
Sure. Habituation, empirically, I would say there are some differences maybe between male and female animals and their age. Generally, what I do is I begin with 15-minute sessions. After the acute window or the window is placed and the head plate is on, I usually do 3 days in a row of 15 minutes to roughly get the animal habituated. And then as I'm waiting for the window to become chronic, I do this at least once or twice per week thereafter for a 15-minute session. And so by the time that I'm at the chronic window phase, this represents about 10 or so habituation sessions of 15 minutes. And I find generally pretty good outcomes. It's also important, I find, when you introduce the mouse to the head fixation any day, that you at least give them about 5 minutes to acclimate just to that immediate change.
Excellent. Thanks so much for sharing. And Rob, do you have anything to add to that? How does it compare with your habituation
It's
protocol?
a fairly similar approach, although we increase the duration each time. So for us, when we're actually doing our experiment, we want to record for about 60 to 90 minutes. So after our head bar surgery, of course, we allow the animals time to fully recover from that, which can take several days or a week. And then we start with a 15-minute habituation, which we then build up to 30 minutes, to 45 minutes, to 60 minutes. But the important thing is it's not just, if they've had 4, they go into the experiment. It's if there's 60 minutes and they seem well adapted to the equipment and they're habituated to that, which, to be perfectly honest, most of them are by the 4 training sessions, but some animals require 5, 6, or 7.
Perfect. Thanks. Next question, Rob. This one's for you. Are graphene arrays compatible with calcium imaging?
Short answer is yes, but it is a little bit complicated. So they're transparent, and the graphene microtransistors are embedded in substrates, and different substrates have different levels of autofluorescence. But we have arrays now that are in a non-autofluorescent substrate, and actually there's a PhD student in our department who is doing combined calcium imaging through the graphene transistor arrays using microscopic calcium imaging. So yes, it is possible.
All right, very interesting. Tony, here's a question for you again about calcium imaging, but is the calcium activity in microglia a product of calcium influx or due to intracellular signaling?
Yeah, that's a good question. So, having not performed the pharmacology to distinguish that, the literature would suggest that there are both ionotropic receptors that can allow calcium into microglia, as well as store-operated GQ type G proteins that can also elevate calcium. In the literature at large, in vivo, it appears that at least damage responses to laser burn or neuronal death, those seem to largely be evoked through an intracellular calcium signaling mechanism and ER release of calcium. But likely the context can also alter which types of calcium influx or store operation might occur.
Excellent. Excuse me. Next question. So you both clearly got some really, really beautiful videos in your presentation. So it seems like head fixation is working really well. But are there any limitations to head fixed mice for your research? And maybe Rob, I'll direct this one to you first.
Well, of course, it's the ability really to record for longer periods of time. So in epilepsy, sorry, there's a bit of background noise. Sorry. In epilepsy, clearly what we'd like to be able to do is record spontaneous seizures. And there are obviously rodent models, mouse models, where you can have spontaneous seizures, but these tend to occur at quite a low frequency, so that your chances of capturing them inside a head fixation apparatus during a 60 or 90-minute window is quite short. And so, I mean, it is possible, and we are doing these studies, but you can sometimes have experiments where you're not going to have a seizure that day. But the good thing about them is that because these are chronic experiments, you can put the animal in several days and hopefully be able to pick up one of those chronic seizures. But it's certainly a lot easier to induce seizures with chemiconvulsant within a 60 to 90-minute time window.
Right, great answer. Rob, anything to add to that? Are there any limitations of head-fixed mice in your research?
And I would add to that, there's always the theory that there can be restraint stress in the animal, but I do feel that's largely alleviated by a proper habituation protocol. For my research, looking at these really optically thin microglial processes, about 1 or 2 microns in diameter, and having to image them with a time series, basically from the same Z-plane, head fixation is like essential. I don't see any way around it. To image up to 90 minutes from a single Z-plane from such an optically thin structure does require a pretty stringent way to maintain the Z level, and head fixation really helps us do that.
Right. Great answer. And Tony, keep you on here for the next question. Where does the 10 to 15 minute latency come from in microglia calcium activity?
Yeah, that's a good question. So in the literature, generally speaking, if you were to increase neuronal activity, let's say with high frequency stimulation, it's been well known for about 25 years that astrocytes, the other glial cell, will have an immediate calcium response. The question for microglia compared to astrocytes is, so why does this take 10 to 15 minutes? And our thinking is that this is integrated into the structural responses of microglia. So if you were to do a focal laser burn, microglia do not immediately arrive at that laser burn. They take about an equivalent 10 to 15 minutes to arrive there. So our hypothesis is that this latency represents that calcium is probably important for the later stages of motility, or maybe even when the processes contact certain structures, such as a lesion or a hyperactive or hypoactive neuron. That contact would occur about 10 to 15 minutes after the activity change based off process structural characteristics. And that's our leading hypothesis about where the latency comes from.
Great answer. Next question is for you, Rob. Where can we get graphene arrays? Is there a commercial supplier?
There may be soon. So the design and initial development characterization is part of a large European Commission consortium. It's the Graphene Flagship Project, which has many partners across Europe. And at the moment, let's say we're at the stage where we're characterizing and actually now using these arrays to answer biological questions. And it's really at the moment accessible by members of that consortium. But I'm under the understanding that multi-channel systems now have this technology and will very shortly be launching a commercially available version of this. So probably check in with multi-channel systems in a few months' time.
Excellent, thanks. And this question kind of goes back to, Tony, what you were just saying about needing to maintain the Z level. Why did you choose the mobile home cage to do this over similar tools like VR or maybe airlifted balls or treadmills?
Right. So a lot of it was actually the home cage system actually existed in the lab before I came there. I had actually never done in vivo recording before, and generally what I find is that so I can't really speak empirically to having comparisons with styrofoam balls or other systems, but I do feel that the flat chamber on a airlifted flat table really does provide you with the least amount of Z-axis movement. And of course, when you have that Z-axis movement, you have chatter in your baseline calcium transients, and that definitely hurts it. So having not a strong experience in all different modalities for imaging, platform-wise, this one certainly theoretically has made the most sense and has greatly helped our ability to look at a single Z-plane for up to 90 minutes.
Excellent. And Rob, anything to add on to that for you?
Yeah, I have used both. I started as using stereophane balls, and I've moved to the Neurotar system. I think the main advantage for me is the ease of which you can bring electrodes or whatever you need in from the side, particularly if you say you're calcium imaging from above and you're wanting to put in a fiber optic electrode, 2 electrodes, an LFP electrode, everything else. It's much easier to have a stable base. You can put these manipulator clamps around the Neurotar frame. In fact, actually, we can attach stereotaxic arms to the Neurotar frame, which allows us to inject our drugs at known distances, etc. So it's just a little bit more practical.
Perfect. Next question here. How easy is it to integrate this setup with other kinds of measurement like video tracking? Maybe Rob, I'll give this one to you first.
Thanks. Well, the way we do this at the moment is we actually have 4 or 5 different, as I say, bits of equipment that are all running at the same time. And we're running these through a CED board to timestamp everything. But I'm aware that Neurotar do have a video tracking system, but that's not something that I've upgraded to just yet. But I'll be looking into it.
Excellent. And Tony, anything to add to that?
Yeah, actually, we recently did upgrade our Neurotar system to also include the magnetic tracking ability. And we've also added an infrared camera into our 2-photon system to also live image mice. We're actually actively in the process of integrating everything, including even a picospritzer board through just a TTL pulse system. And all of it can be simultaneously started through our 2-photon imaging software. So we're actively actually being able to integrate both the ability to track location, video record, and even introduce stimuli all through a single trigger. So yeah, this system actually can be very helpful for integrating a number of modalities.
Excellent, great answer. And I think in the interest of time, we'll make this next question the last one. Do either of you have any experience working with rat models? Do you know if this methodology and whether these technologies are applicable, can be used with rats?
I personally haven't tried head fixation in rats. I'm not aware of people who do. Rats tend to be an awful lot stronger than mice. and I think the head fixation would have to be particularly well secured, particularly for an epilepsy-related question. For those reasons, I've stuck with mice so far.
Excellent. Well, great. Thanks so much, Rob and Tony, for all your fantastic insights today, both in your presentations as well as this Q&A session.
Thank you very much.
Thank you.